So what I'm going to do now is I'm going to give you a complete overview over aerobic cell respiration, and there are several steps involved, and it might take us a little bit longer, so I guess it's going to be a slightly longer video. And my advice is you just copy along the stages. I'm going to explain all of this, it's basically a question of simply memorizing the whole thing. Of course, you have to understand what's going on, but I think there's no way around also memorizing the different stages. I just give you an overview first. The first stage is called glycolysis. Then I'm going to talk about the so-called link reaction. The next one is going to be the Krebs cycle, and you can see that I'm going to fill in all of these spaces here. And then the last stage is oxidative phosphorylation, and that's what I'm going to explain you now. Okay. My advice is that you probably have listened first to the cell respiration of the standard level, because this kind of gives you an overall overview. But now we take it a step further and we go into more detail here. So the first step is glycolysis, and remember in cell respiration I'd like to explain how glucose is broken down, and I'm going to give you the overall formula, and then we're going to break it down further. So what we have is the following. We have C6H12O6, that's glucose. C6H12O6. Glucose is our main energy source. We need oxygen, 6 molecules of oxygen. We get it from the air. And this will result in a complete breakdown of glucose. So we make 6 CO2s. We exhale that. That's what we breathe out. And 6 molecules of water, 6 H2O. And what's the whole purpose of this? This is because in the whole process we make a lot of energy. And I indicate it like this: ATP, adenosine triphosphate, but we cannot simply make ATP out of nothing. We have to add ADP plus inorganic phosphate. And I'm not it looks like it's only one ATP is made. No, many. Okay. Around they're not quite certain, the scientists, but around 38 ATPs. 36 to 38 ATPs are made in the process. Let's say 36 net ATPs. 38 total, but because we use 2 ATPs in the beginning, it makes a 36. So maybe 36 would be actually the better one. It depends a little bit how you define it. Okay. But I read as well that scientists are not quite certain yet, and they're doing their math, and it's somewhere in this area. It doesn't really matter, per molecule of glucose. And what I'm going to do now is I'm going to I've drawn this here as one single line, one single step. But in reality, there are many steps, and the steps I just explained to you, glycolysis, link reaction, and so on. You can find them in here, and I'm going to simply trace you through these stages. Okay, so that is the overall formula of aerobic cell respiration. I'm just going to erase everything right now again because I need the space. Okay, so let's get started. And we always remember that we have to keep track of our carbons here. Okay, we have. Let me talk a little bit more into the microphone. I'll have to move this over. Maybe you can hear me now a little bit better. So, and what I'm going to do is I'm going to start off over here. I'm going to start here at the beginning with glucose, of course, and I'm just going to write glucose. But I'm going to also keep track of the carbon. So I'm going to say it's a C6 substance. Okay. And that's what we start off with. And if we start with 6 carbons over here, we have to have over here at the end, we also have to have 6 carbons, right? So, or at least in the whole process, we have to have the same amount of carbons that we add here at the beginning, we have to have at the end. So, first of all, the first step is the glucose is too stable and it doesn't want to react. But we want to break down the glucose to release the energy. So what we have to do is we have to add energy in the form of ATP. And what we're doing is we're adding 2 ATP, and we're transferring the third phosphate, because an ATP has 3 phosphates, and the third phosphate we're transferring to the glucose, and we may have 2 ADPs. We remember that an ATP has an adenosine phosphate, and over here a squiggly line that indicates energy, the third phosphate. And what we have here at the end is we have the adenosine, and we have 2 phosphates, diphosphate. What happened to the third phosphate? It got attached to the glucose, and what we have then is we have a 6 carbon sugar with 2 phosphates attached. So it's okay, that is not good. So we have over here hexose, and then this hexose has is called biphosphate, and I'm just going to indicate the P like this to indicate a phosphate. So it's still a 6 carbon, it's a C6 substance, okay? And this first step is called phosphorylation. So you might want to write this down, maybe somewhere it's supposed to be a 1. Phosphorylation. We're adding phosphate. The phosphorylation, and this is an energy investment. And those of you who are in chemistry, what we're doing is we're adding activation energy here, because otherwise it wouldn't want to react. Okay. The idea is to get more energy out, but here we have to put more energy in first. We have to put some energy in first to get more energy out later. It's like striking a match. You have to strike the match first. You've got to add some energy first before you get more energy out later. Yeah. But we still have our 6 carbons over here, and this one is unstable. I mean, that's the whole. That's why we added phosphate. Okay, that's unstable. And what we do is the following. I'm just going to make a drawing over here. The glucose, every little dot is a carbon. Carbon 2, 3, 4, 5, 6. That's my glucose. Hexose biphosphate has a phosphate group attached. Carbon, 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 and the second phosphate. Okay, so that's a little bit how you can imagine the whole thing. As a consequence of this, because it is unstable, it's going to fall apart. It's going to fall apart into two parts, and this is called lysis. And now I don't have space, so I got to write it over here. So step number 2. Maybe when you actually copy it into your notebook, it would be a good idea to have all the labels on one side and not just go back forth. Okay, like I'm doing here. It's called lysis. Lysis is splitting, and what happens here in the lysis process? This is supposed to be the lysis step. Let's write it closer to this orange arrow. No, now it looks like I'm adding lysis here to the equation. I mean, you get the idea. I'm just going to label it over here. Number 2, lysis. Lysis. That's lysis stage. And what I have is it falls apart, and I have 2 triose phosphate molecules. 2 triose phosphate, and it looks like this. We have a phosphate here and 3 carbon, and then we have got the second one over here. So you see it falls apart, and this splitting is called lysis. Yeah, that's the whole point. We wanted to make it unstable. It falls apart, and now we have the following situation. Now the third stage is oxidation, and I write this numbers here. Stage number 3. So oxidation. And now we get energy out. Okay, and how do we get the energy out? We get energy out in the form of high energy electrons. So the triose phosphate molecules, they now are ready to give off high energy electrons. The question is, where do they give it off to? And we do have a so-called an electron carrier molecule. It's called NAD+. So we have 2 NAD+, 1 NAD for each triose phosphate, and it's supposed to be a plus over here, and we make 2 NADH. As I already talked about this stage in the introduction, and we're removing electrons here. So that is an energy harvesting stage, and still we would like to now also make ATP. And what we're doing right now is we have another energy harvesting stage, and this is called number 4, ATP formation. ATP formation. And what we're doing right now is we're adding to four ADPs, and we're getting out 4 ATPs. So in other words, what we have is the following. The triose phosphate, they contain these, here's a phosphate and here is a phosphate, and they get transferred to the ADP. Okay, and to make ATP, so we're getting more energy out because we're making ATP. And those of you who are really careful, you will notice, well, actually, don't we need 4 phosphates, 1 for each ADP, but we only have 2 over here. And the reason is because we're adding 2 inorganic phosphates, just for those of you who are worried about the stoichiometry here. Okay, so we get 2 phosphates, we're adding 2 extra phosphates, we get 2 phosphates over here from the triose phosphate, and we have then we make 4 ATPs, and at the end, a very important molecule, we have 2 times a pyruvate. Pyruvate, that's supposed to be a V over here, and it's 2 times a C3 substance. Okay, and that is the how shall I say, these are the stages, and what we have over here, this is the energy harvesting stage. So we're getting energy, more energy out. Energy harvesting. And overall we have the following situation. We have 1 glucose, and what we're making is we're making 2 pyruvate. We're gaining 2 ATPs. We're making 4 here at the end, but we've invested 2 over here, so that means we have a net gain of 2 ATPs. And what we also have is we are making 2 NADH. So that's where our energy is. So we have energy in the NADH, we have energy in the ATP, and we've broken down glucose, right? So we have a 1 C6 molecule over here to 2 times a C3. So the number of carbons must stay the same, and we got some energy out. That was glycolysis. If they ask you a question here, what is the correct steps, a sequence of steps? Remember, phosphorylation makes it unstable. Then we have lysis, we have oxidation, and ATP formation. These are the stages. I would say way too chaotic here on the foil. Please write it down properly so that it's clear, the different stages are clear. Okay, so that is, now we have this, and where does this occur? In the cytoplasm of a cell. Okay, that is in the cytoplasm of a cell, and has the advantage that you do not need oxygen. Excuse me, you don't need oxygen, but you don't make a lot of ATP. You don't release a lot of energy in this stage. So we move on. Okay, so what happens to the pyruvate? The pyruvate now goes into the mitochondrion, and this is now the link reaction, and this link reaction takes place in the mitochondrion matrix of the mitochondrion, mitochondria plural. Okay, and of course we have to start over here with a pyruvate. Okay, and the C3. Now, yes, per molecule of glucose we make 2 pyruvates. I'm just going to look at one of them here. So it's going to happen 2 times, 1 for each pyruvate molecule for each glucose. Okay, and what we do now is the following. It's we are now taking CO2 away, and we're oxidizing. How do we oxidize pyruvate? By removing electrons. How do we remove electrons? By adding NAD+. Okay, we're adding NAD+, we're getting out NADH. So you notice that it's a neutral molecule, so we're transferring an electron over here. Okay, I mean, the thing is the following. If you want to, it's easy for you to memorize if you just say we make NADH. If we make NADH, then you know that we have to add NAD+, so it's not necessary to also draw this here, because it's kind of clear. Yeah, so that is, we're oxidizing, but there's something else that happens in this stage. What we're doing is we're also removing a CO2. Yeah, because we're exhaling CO2. Remember, we're producing CO2 when we break down glucose. That's one of the CO2s, and we're also adding something that we call a coenzyme A. Coenzyme A. Okay, in this step, and then we have at the end acetyl coenzyme A. That is the substance, also a very important central substance that you have to know. And this step, because we're oxidizing and we are decarboxylating, we call this step an oxidative decarboxylation. In other words, if you just remember the link reaction is oxidative decarboxylation, then you already know that we have to oxidize. How do we do that? By adding NAD, by making NADH, and we're taking the CO2 away. Yeah, and we have to add coenzyme A in this step additionally. Now the acetyl group over here is a C2 substance. Well, because 2 plus 1 makes 3. Yeah, so again the stoichiometry has to be correct here. Now that is the link reaction. You can also call this oxidation of pyruvic acid, because pyruvate or pyruvic acid, it's more or less the same thing. Okay, so you can also call this this whole step, the link reaction, it can also be called oxidation of pyruvate or pyruvic acid. Okay, pyruvate, oxidation of pyruvate. Of course, we're oxidizing over here. Yeah. So for your information, the acetyl-CoA can be made now because before that in the glycolysis with glucose we make pyruvate, now we have acetyl-CoA. This acetyl-CoA can also be made in a different way by the breakdown of fatty acids. So lipids, yeah, acids. Can also make acetyl-CoA. You have to know this, I'm just telling you. So fatty acids can only be broken down over the aerobic pathway. Higher level folks have to know this. Now you know, if they ask you multiple choice question, how can fatty acids be broken down? Well, in aerobic respiration, and they're converted to acetyl-CoA. And if I'm just mentioning this very quickly, if you have a fatty acid over here, like that's my fatty acid, and we must have an even number of carbons. It's really important, otherwise it's not it's poisonous. And what happens is the following, that always two of them are going to be converted to acetyl-CoA. So we can make 3 acetyl-CoAs out of this fatty acid here. Okay, side information. Okay, but no, you have to know it. There was once a multiple choice question that asked how many acetyl-CoA and how many acetyl-CoAs can you make of this fatty acid? Yeah, and 2 carbons of course always make an acetyl-CoA. So that is the link reaction. Okay, so we remember in the link reaction we convert pyruvate to acetyl-CoA. Okay, and now let's move on to the next stage, and that's a huge one. It's called the Krebs cycle, or it's also known as the citric acid cycle, or Krebs citric acid cycle. Okay, and what shall I say? I mean, all you gotta do is you gotta memorize this. Yeah, we start over here with acetyl-CoA. Coenzyme A. Okay, and what happens here is the following. This is a C2 substance. The acetyl group at least is a C2 substance, and what we're doing is right now is we're combining the C2 substance with a C4 substance called oxaloacetate. You gotta know this. Acetate or oxaloacetic acid, and that's a C4 substance. Okay, and when you combine a C4 substance with a C2 substance, you make a C6 substance, and this C6 is called citric acid, and has 6 carbons again. Okay, the coenzyme A we have to remove here. Okay, so we're just taking this out. Coenzyme A, yeah. So we just look at the acetyl group, and we combine with oxaloacetate to make a C6 substance called citric acid. So that's the first thing that we're forming in the citric acid cycle. That's why it's got its name. Okay. So and now what happens next? And now the point of the Krebs cycle is to completely break down the 2 carbons that we've added to release energy. So what we have over here in this first step is we're going from a C6 to a C5 substance. What do we have to do to go from a C6 to a C5 substance? And what do we have to do to get energy out? We have a step that we call, surprise, surprise, oxidative decarboxylation. So what are we doing? We're making NADH, and we're producing CO2. Don't we have to add NAD+? Yes, of course. I didn't draw it because it should be clear. Okay, every time when I make NADH, I have to add NAD+. I mean, you already know that, so I'm not going to draw it, right? Oxidative decarboxylation. Over here, from a C5 to a C4 substance. How do we get from here to over here? What do we have to do? Oxidative decarboxylation. So we make NADH, and we're taking out CO2 at the same time, and now we're at a C4 substance. So we've added 2 carbons over here, and we've taken out 2 carbons over here. Okay, and this C4 substance over here is now still energy rich, so we can still get some energy out. And what we're now doing is this C4 substance can now be used to make ATP. So what I'm doing is the following. I'm making now ATP. Now you're going to say, well, how do I make ATP? Well, I have to add ADP, of course, plus an inorganic phosphate. Yeah. And where do we take the energy for this? Well, the energy is in the substance over here. Okay. Well, we have a C4 over here. We still have a C4 substance over here because we didn't take any carbon dioxide out. But it's still energy rich. So what we can now do is we can remove electrons, an oxidation step. And now we're not using NAD+ to oxidize, but a similar substance called FAD. FAD. And we have FADH2 now you're going to ask, okay, what is the difference between FAD and NAD and FADH2 and NADH? They're both energy carrying molecules, but the FAD does not carry as much energy as the NAD. That's all, okay? But we remove it's an oxidation, okay? Oxidation, then over here we have ATP formation, okay? This is oxidative decarboxylation, okay? And this one, of course, is also oxidative decarboxylation. Okay, bad handwriting, okay? So we have a C4 over here. We're taking electrons away. How many carbons do we have over here? Well, we still have a C4 substance. We didn't take any carbons away. Yeah? What do we now? How do we get from here to over here? Well, we have another oxidation step, and now we're making NADH. We're again removing electrons, so it's another oxidation. Okay. So what you've seen is the following. We've now completely broken down. And we're back at oxaloacetate. Okay. So what we've done is we've completely broken down now whatever we added. We added 2 carbons. We removed 2 carbons in the form of CO2, and we've also removed a lot of energy over here in the form of high energy electrons. Over here in the form of high energy electrons. Directly in the form of ATP. Here we've got some energy. We've got here some energy in here in the form of high energy electrons, and the NADH over here. We've also got some energy here in the form of high energy electrons. Wow, we got all the energy out, and it's now captured directly in ATP, which is kind of cool. That's useful, but also in the form of NADH and FADH2, and that is basically where we have a plus ATP. This is where our energy is. Okay, so that is cool. The ATP is fine. But now we have a problem. What about these 2 here? That's not useful. We somehow have to convert this to ATP, the energy, and that's going to be the next step. Okay, so to summarize this, in the citric acid cycle, we're completely oxidizing all organic substances. So there is no carbon left anymore, and the CO2 that we've added over here. No, not added over here. We've added over here. We get out over here, and the energy which was stored, we got out in here in the NADH, FADH2, and in the ATP. Okay. So, for your information, this is a pretty important cycle because also, for example, when we eat proteins and so on, they're all fed in somewhere into this cycle. All the other organic food molecules that we're eating are fed in here in the cycle as well and completely burned. So that's the Krebs cycle. Now let's move on. Next step is we want to convert the high energy electrons into ATP. Oxidative phosphorylation, also known as an electron transport. Electron transport. Electron transport chain. Where does it happen? In the mitochondrial membrane, in the inner membrane. Mitochondrial membrane, but the inner one. The mitochondria, they have 2 membranes, and that's the membrane over here. So you can imagine over here I've got my phospholipid bilayer. These are supposed to be the phospholipids, right? Yeah, these are all the orange things here. These are membrane proteins. And what we have over here is this is the matrix, and that is the intermembrane space of the mitochondria. Yeah, so that's the inner membrane, and of course down here, all the way down here, that's the actual mitochondrial membrane. Okay, pretty large over here. And what we want to do is the point of oxidative phosphorylation is somehow to convert the high energy electrons of NADH and FADH2. We somehow want to convert this to ATP because that's the useful thing. Okay. So now let's have a look where that was my mobile phone. Let's get started over here. We start off with the NADH. So all of these orange, the first 3 orange proteins that you see over here, these are transmembrane proteins. They're electron carriers. Okay. So what we do is the NAD, this guy over here, okay, goes to the first electron carrier, NADH, sorry. We got the NADH from the Krebs cycle, and it goes to the electron carrier and gives off its electrons, and then it makes again NAD+. Okay, so it becomes it transfers the electrons. So this orange protein down here, the first electron carrier, becomes reduced. Okay, and now the electrons now are passed on. So that's supposed to be my electron. Is passed on now from electron carrier to electron carrier. So I'm just going to write E minus as electron, and it's going to be it's passed on again over here. Okay, so the electrons. And as this happens, as it is passed on from electron carrier to electron carrier, the electron loses energy. It loses energy, but you got to ask now the question, well, where does the energy go? It cannot simply magically disappear. And the energy of the electron is used to pump, active transport, to pump hydrogen ions across the membrane. So what we have is we're pumping hydrogen ions across here, and what we have is we have a lot of hydrogen ions, protons, a hydrogen ion, that's a proton, accumulating down here. Yeah? It's a form of active transport, but an active transport which uses the energy of electrons. Normally, I don't know if you've already studied that chapter. Normally in pump membrane pumps, they use ATP as an energy source, but here we have those that use electrons. And so the energy, the electrons lose energy as it is passed on, and this energy is used to kind of build, but to build up hydrogens down here. And this is what we call a PMF, proton motive force. It's supposed to be an F, okay? The proton motive means movement. Yeah, so they don't want to be here anymore because they want to diffuse back, because we have a high concentration, high H plus concentration. And if you have a high concentration on one side in the intramembrane space and a low concentration on the other side, okay, what is going to happen? What do they want to do? They want to move to the other side. Can they do that? Well, yeah, there is something, this little thing over here that we call an ATP synthase. ATP synthase. And the ATP synthase makes ATP. But for making ATP, you need energy. Now, where does the energy come from? Well, the energy comes from the diffusion of the hydrogens back into the matrix. So the hydrogens, when they're going back, they're diffusing back because we have a high concentration down here, and they're moving to a low concentration, and this releases energy. And this energy can now be used to attach ADP, an inorganic phosphate, to make ATP. Remember, I told you, how do we make ATP over here? Well, that's how we use the energy from the protons from the hydrogen ions that stream back. Okay, and so we make a lot of ATP here. Okay, now I'm not quite finished yet, because what happens to the low energy electron that we have here? We still have an electron over here, which has given off most of its energy, but the thing is we still need to do something with it, otherwise, yeah. So, and what we now need is we now use a molecule, for the stoichiometry, it's like this, that half a molecule of oxygen, and we're making water, H2O. And for that, we need the electron. So the important thing is that oxygen, if you want, you can say one O2 makes two H2Os, but then you need more electrons. But the idea is the following, that the oxygen over here is our final electron acceptor. And now you understand why we need oxygen, because if we don't have enough oxygen, then our cells cannot do aerobic cell respiration, then they cannot produce ATP, and the cells die because they don't have enough energy. So the oxygen that we inhale, that we breathe, is there to release energy. Otherwise we have an energy problem, or the cells have an energy problem. So the oxygen is our final electron acceptor, and now we're making the water. Okay, and that was it, at least all of the stages. Okay, so I know this was quite a bit of how shall I say, quite a lot of information, but I just remembered something that I didn't explain you yet. Okay, I gotta do that. I don't know, I'm going to make a separate box over here, okay, to simply say this. There are two ways of making ATP. We can make ATP or using the ATP synthase, okay, and that is called oxidative phosphorylation. So ATP can be made by oxidative phosphorylation please write it out. Don't just, yeah, but it's, you got it up here, right? So, yeah, so please write it out. And the second way of making ATP, so we need a membrane here, right? And the ATP synthase and the membrane, and we need a proton motive force, yeah, and high concentration of hydrogens over here. And that's one way of making ATP, but there's a second way of making ATP, which you already know. And the second way of making ATP is this one over here. Yeah, do you see this? Okay. Where you do not need a membrane, because I also didn't tell you the citric acid cycle occurs in the matrix of the mitochondrion. Yeah, so we don't have a membrane, and this way of making ATP is called substrate level phosphorylation. Relation, a little chaotic, I admit. Okay, so these are the two ways of making making ATPs. So substrate level phosphorylation, for your information, is the second one. So that was, remember, this is one way over here. That's one way of making ATP using substrate level phosphorylation. And in case you forgot, look at this over here, ATP formation here as well. In glycolysis, you also have substrate level. You have also substrate level ATP formation, right? And that's it. Okay, so these were the main stages over here. Yeah, and please take notes of all of these things. Use a lot of space. Make it nicely to not as chaotic as I'm doing it here. Okay, so simply that you have a better overview over the stages, and then all you do is you memorize the whole thing and memorize what goes in, what goes out of the stages, and also memorize the number of carbons. I forgot something. I just realized. Oh, and you thought I'm finished. What about the FADH2 over here? Okay, FADH2. I think I already told you that it doesn't have enough energy, not as much as NAD, so the electrons go over here. They go in here. That's it. Okay. The FADH2 also transfers its electrons, but not into the first one, but only later on, because it does not. The electrons don't have as much energy. So, and now I'm really finished. Wish you all the best.